On the one hand, if this facilitates covalent bond formation between the two pieces, it could stabilize the complex, and it could help. But on the other hand, dangling cysteines can give rise to unwanted aggregation, or perhaps it could cross-link to an incorrect protein that also had a dangling cysteine. And that would then hurt our reconstitution. So to test whether the role of these cysteine residues, we prepared a couple mutants of the A and B fragments. We mutated the cysteine residues to either an alanine pair or a serine pair, with the thought that a hydrophobic contact, as with an alanine pair, or a hydrogen bond, as with a serine pair, might compensate for the loss of stability that comes with removing these cysteines. And when we tested them, we found that the parent system that had both cysteines intact gave bright fluorescence, but both of these mutants completely abolished the signal. And furthermore, we even tried adding an N175S mutation, which is known to improve the stability of full-length HRP, in the hope that this would compensate for that loss of stability, but it still gave no signal. So from this, we conclude that these cysteine residues are crucial for our system, and therefore, in this image where we have activity, we can conclude that an intermolecular disulfide is formed between these two pieces. So what I've shown you here is that within the ER lumen, these pieces can reconstitute and fold into an active form. But I mentioned that the ER lumen has a lot of chaperones that assist in this process. And to apply these fragments for synapse detection, we would need them to reconstitute extracellularly, which is a much more demanding application because it's not known whether any chaperones reside in extracellular space that would facilitate this type of disulfide bond formation. So a crucial next step was to test whether these could come together outside the cell. So to do that, I'm going to introduce to you a model experiment that we use, and which I'll show several times throughout the talk. Ideally, we would like to test this in neurons, which we can do, but it's quite a bit harder to work with cultured neurons. So as a model system, we used HEK cells, which is an easy to work with cell line. And we constructed two constructs. One had the A fragment fused to Nerexin, the other had the B fragment fused to neuroligin. And these are the synaptic proteins I introduced in the beginning that interact across the synaptic cloud. In one pool of HEK cells, we transfected with the Nerexin construct and a blue fluorescent marker, and in a separate pool, we transfected with neuroligin and a green marker. And our hope is that if we put these two pools of cells together, we should see HRP activity reconstituted specifically at the contact sites. So this is showing you the actual data. Here you can see an image of the green cells. Here is an image of the blue cells. And in this merge channel, you can see one contact site right here. And we are very happy to see that HRP activity, as visualized with our biotin phenol substrate, which gives spatially restricted labeling, was indeed localized to this contact site. And I've drawn a large cartoon here. This is a depiction of these two cells, just to show you more explicitly what we're looking for. So this was surprising. This implied that these fragments really could reconstitute in between two cells. But one big caveat of this is that heme supplementation was required. In order for this to work, I had to add heme to the media, and when heme was omitted, this fluorescence was essentially not detectable. So next we moved on to a more relevant experiment, which is testing the same thing in cultured neurons. Here, the A fragment and B fragment were introduced into two different pools marked by blue or green fluorescent proteins. And again, we're very happy to see that HRP activity could be observed specifically at these contact sites. So now we're getting closer to our actual experiment of mapping synaptic connections, because this is a more relevant type of cell. Again, though, this came with a caveat of heme supplementation being required. An important question in all of this is, our thought is that the reconstitution should be dependent on the interaction between Nerexin and neuroligin. But if it turns out that our two pieces come together spontaneously, that would be a serious problem, and we wouldn't be able to really claim that we're detecting synapses. So to test whether this is true, shown up here is just another field of view for the constructs I presented to you earlier. Here are some deletion views where now we're just displaying the two fragments on their own on the cell surface of two pools of neurons, and Nerexin and neuroligin are removed from this. So now, in the absence of this protein-protein interaction, we see no fluorescence. And so this gave us confidence that we had a method that truly was detecting synapses, and not just an artifact of these two fragments coming together. So now that we are convinced that this was working for synapse detection and it was specific for synapses, we performed a side-by-side comparison with split GFP, the method that we are hoping to overcome the limitations of. Shown on the top is our data with split HRP. You can see bright fluorescence corresponding to the expected contact sites. In this row, we have split GFP. The two pools of neurons are marked in blue and green, and false colored in red is the GFP fluorescence to facilitate a side-by-side comparison. And you can see that there's no detectable signal at a matched intensity scale. when we raise the contrast to the DFB fluorescence, you can start to see some fluorescence that overlays with the expected contact sites. So this establishes that this system worked in our hands, but it was just much, much different than split HRP. So this was very exciting and encouraging, but again, note the caveat that heme supplementation was required. So I've been mentioning several times this need for heme supplementation. And we need to think a little bit about how exactly is heme getting into the active site of our reconstituted complex. First of all, this enzyme has no activity at all without heme, so we can be basically sure that heme got there somehow. And there are two plausible mechanisms. One mechanism is that the two fragments are trafficked through the secretory pathway, make it to the cell surface, and one of the pieces, let's say the B fragment, picks up heme along the way. Then they meet in the synaptic cleft, the enzyme reconstitutes into an active form, and heme is already present because it came along with one of the pieces. This is one possible mechanism, but another is that the fragments may traffic to the surface, but neither binds heme. They meet in the synaptic cleft, fold into an active form, but lacking the heme cofactor. And it's only upon the addition of exogenous heme that it diffuses into the active site. So the data we have so far, along with other data I didn't have time to show, supports the idea that this is the predominant mechanism by which our system is working. And this represents somewhat of a drawback, because although it's easy to add exogenous heme to our cultured cells, it's quite challenging to deliver heme into tissue where there are a lot of cells and small molecules don't diffuse very readily into that tissue. So despite this potential concern, we were encouraged enough to give this a try in the mouse brain. We wanted to see, could we really use our synapse detection method to map synaptic circuits in a more relevant physiological system. This work was done in collaboration with Masa Yamagata and Josh Sains at Harvard, and I'll give you a scheme of what we tried to do. This is a very crude picture of the mouse brain, and shown here are the approximate locations of the retina and the superior colliculus. And it's known that neurons in the retina extend their axons all the way to the midbrain to form synapses with neurons in the colliculus, and these synapses are involved in visual processing. So this served as a positive control. In other words, this is a set of neurons that we knew formed synapses in a specified location. So Masa prepared viruses encoding either the A fragment or the B fragment, injected them into these two spatially separated locations, then sacrificed the mice, dissected out the brain, cultured the brain in heme-containing media to try to get heme in there, then fixed the brain chemically, cut sections of the superior colliculus, and checked for synapses. So I'm skipping a lot of details of that, but I'll show you some representative fluorescence data which gets to the main conclusion. So this is fluorescence imaging of a section of the superior colliculus. This is where we expected these synapses to form between our two sets of neurons that we infected. The two pools of neurons here are marked with either blue or red. HRP activity is shown in green. And pointed out by these white arrows are sites where we do see puncta of HRP activity, and these overlay specifically with contact sites between our two pools of neurons. So this actually was somewhat of an encouraging result and implied that we are detecting synapses in the mouse brain, and we were convinced that it was specific. But you may notice that there's a big problem with this, which is that the signal-to-noise ratio is very poor. You can see this fluorescence above the background, but it doesn't stand out by much. And furthermore, the requirement for heme supplementation was inconvenient, and Masa felt that it wasn't very successful. It didn't penetrate evenly into the tissue, and so there are regions of the brain that just weren't labeled very well. So this was disappointing because we weren't confident that this would actually be useful for a number of applications in mapping synaptic circuits beyond this proof of principle experiment. So we had to ask ourselves, how can we make our split HRP reporter system better? A typical way that people do this when faced with this problem is to try a different cut site, but we did not take this approach because we had already been pretty exhausted initially, and we didn't feel optimistic that any other cut site would give a substantial improvement. So the approach that we turned to was to perform directed evolution on the A fragment of HRP. This is to make a library of random mutants of this fragment, and then in the hope that one of the mutations will have beneficial properties, and then try to select out mutations that give rise to an improved performance in our final application for synapse detection. And I will note that we wanted to do evolution on this A fragment because, first of all, it's bigger, so there are more places where beneficial mutations could occur. And second of all, most of the heme binding residues are located on the A fragment, and we hypothesized that heme binding would be a crucial parameter to optimize. The strategy that we selected was yeast display evolution. And it's important to note that in yeast display, you traffic a protein of interest to the cell surface through the secretory pathway. This is a crucial consideration because HRP fails in any other compartment. So that's why this method was suitable for what we wanted to do. And I'll outline briefly the workflow that's involved in this. First, we needed to create a library of yeast cells, all displaying a different mutant of this A fragment. Then we needed to somehow append a fluorescence label to the cells displaying the most active mutants. Then isolate the most active cells using flow cytometry, sequence yeast DNA to determine if there were any promising mutations that were converged upon, and then finally, all of this needed to be validated in mammalian cells because this yeast display platform is not a perfect simulation of what we really wanted to do. So there was a very important next step, which was to actually follow up on this in the mammalian cells. So this is an overview of what I'm going to show you.